We'd like to invite the speakers from the first session to come up, as well as Florian and Shay to come up on stage, and we'll have a panel discussion now. Maura and I have prepared some questions, but if there's any questions related to those presentations now, specifically, we're happy to take them as the panel members come up. So the presenters from the first session sit on the right, and we have two chairs over here for Florian and Shay, just so we're
We
clear.
keep them separate or what?
It's just easier, because it's a way to fit on the...
And we hope that Oliver is still with us. That was great that you could share this interesting data now also on the Delta in chronic hepatitis D patients. So let's see. It's a hard time in Europe for him to be with us, but
You?
Yeah.
Me? Start.
you can start.
Okay.
Whatever.
Yeah, I mean, I think you saw it there. Clearly reflects one of the challenges we have, and there's a lot of heterogeneity in the response and heterogeneity sort of across either the disease phenotypes or the drugs that are being used. And I guess, here I think it's all speculation and opinion for most of this at this point, right? Because we're still waiting for translational data to come in, but really trying to understand, when you saw some of these follow-up times of, many, many weeks, months at a time, I guess I'd like to get some input from the audience and sort of hear as well. It's very unlikely that the drug is hanging around that long. So, it almost, in some of these cases, anti-S is associated with the sustained response. But I think really what this is telling us is, the functional cure patients that probably almost certainly still have some virus in the liver, and there'll be some of these therapies that prevent that, just basically control that. And so I'd like, speculation on what are those mechanisms? Why isn't the rest of the liver being infected when we have these therapies being withdrawn, and some of these patients are not anti-HBs positive? We see a very slow rebound, even though the virus is present. You think typically we see a logarithmic infection curve with HBV. Does anybody have any thoughts? And maybe I'll start on the research side down here first. Are hepatocytes just not being reinfected after they take it away? I mean, what is the resistance in some of these cccDNA silenced cells, maybe? Let's start with that from Lena, maybe.
Well, of course, I could imagine that some of the cells, when they don't get reinfected, they maintain their cccDNA silencing, and the viremia is very low. Most of the patients are NUC suppressed or have been suppressed through the treatment, so I can imagine that this is one part of the mechanism. I don't know if the hepatocytes are actually resistant. I don't think we can say that.
Do we know if silenced cccDNA can actually prevent superinfection? I think you showed that, right? You can still superinfect.
Well, I showed a study from Van Hoy Lee's lab showing that cells with an X minus, so an suppressed cccDNA, can get superinfected and reactivated in cell culture.
So that puts the question that how long should we help the situation and protect the hepatocytes from new infection events? So how it's important is to have a profound suppression of viremia, and so in order to protect this reactivation. Is it really key? Can we achieve a more epigenetic suppression? Do we need to get rid of the cccDNA? This is going to be enough to silence for the mechanism that we just try to get some data on to avoid a rebound, especially considering that we have to see what's happened on the immunological response, right? I think that's so.
Yeah, well, we clearly don't have to completely get rid of the cccDNA, because that doesn't happen in a cure of an acute infection. There's that issue going on there. So I don't think we need to get complete elimination.
Not complete, but
Yeah.
probably
Yeah, it's low. The other thing to think about is the best estimates that I've seen on the half-life of the cccDNA in human beings are shorter than what has been believed in the past. This is not an infinitely stable molecule. And so I think in some of these patients, we are getting to the point where natural cccDNA decline is not, you know, the main cause of this, but I think it's starting to contribute to it. So.
I didn't see who was at the microphone first. You guys can fight it out.
Just on this point, this discussion point, I think this is part of the thinking that I had for my question for Alan during the keynote speech. When you have e-antigen negative patients with viremia and you see in their liver you've got one cccDNA per 1,000 cells, it's happening in the natural condition, and I don't understand what's controlling that, and we should try to find out, and I think that might be relevant to what's happening here.
Luca?
Well, just to remind everybody, so the liver has a space to have, at the peak of an acute infection, up to 10 to the 12 genomes per ml. And in naive chronically infected patients, you can have 10 to the 6 viral genomes per ml constantly. So clearly, a small percentage of hepatocytes remains productive in surface antigen. It doesn't matter for CCC integration, without having all the other hepatocytes supporting infection. So I don't have other explanations than what Alan told us. There must be cells that remain over time refractory to infection. There's no other explanation, mathematically speaking. So that's the fact to me. So then if you have 0.1% of hepatocytes that express surface antigen, doesn't matter from CCC or integration, and you kill them off, you're not going to see ALT. You're not going to see a flare. There are just too few of them. And so over time, they may just decay. So I think the mathematics is telling us that the number fits if you can kill them off or cure them, for that matter. But they're just most likely most chronically affected people, especially those with low surface antigen, those that respond the better. A very small percentage of hepatocytes that are infected.
Yeah, I mean, I think, you can see it now with the new models. I think you're right onto something, and this is one of the questions that I've had recently as well, like this resistance. Why are these sort of other hepatocytes resistant? Are they just not becoming infected? And now these infection models with the single-cell data and the transcriptomics is allowing us to start investigating those in primary human infections. I think this is a very important point when we think about samples coming from the clinical studies with the siRNA and the antisense oligos. Antonio?
Well, I don't know, actually, because my question is a little bit off, but if I can ask, because unfortunately also in 20 minutes I have to go.
Me too. Go ahead.
No, the question is really for this data of GSK that, we see many patients that are responding. As you said, your antisense can act dually, let's say, as an antisense to block, let's say, S-antigen replication, but also as a TLR8 agonist. Now, my question is very specific. I don't know whether you might answer or not. So my curiosity is, we have been already other companies that have used pure TLR8 agonists. Can we have a sort of quantification of the quantity of your compounds as a TLR8 agonist and the quantity of the compounds, for example, of Gilead that was used? Do we have an idea whether you're using much more or much less, or is completely different action?
Thank you for the question. We do have preclinical data, which shows a very similar cytokine profile to selgantolimod. So you get the interferon gamma, the IL-12P40, the IL-1RA, and that's been validated in humanized TLR models, where we do see the IL-12P40 signature only. Interestingly, with Bepi, which is unconjugated, we see a greater proportion of that cytokine versus the GalNAc version of ASO. But further work is undergoing, and we're hoping to present some of that preclinical data at the end of the year at ASLD. So the team is definitely working on that.
Okay. So your it seems more potent.
Well,
as a TLR rate. Okay, thank you.
Can I? I'd come back to a little bit what Luca referred to about the liver, which is a huge organ, and as he said, there are tons of cells. And right now, the discussion focuses on how do we get rid of all the active cccDNA and all of this. And that's really going into fine details. But maybe, and that's much harder, I understand that. I mean, the bigger problem is what level of suppression or clearance do you have to achieve that you don't have any problems 20 years down the road? And I mean, we know now in HCV that these are problems that despite treatment and whatnot are possible. So I don't know how to address that, so that's basically, I put that to you, you know. I go in retirement and I'm done.
Yeah, so I mean, I like that. And I actually have a follow-up question for both Shay and Floria on this, right? So, you know, how relevant do you think anti-HBs seroconversion is for the sustained response? Because you've both seen it in the Be Clear and in your study. So what are your thoughts on the relevance of anti-HBs as an important role for durable response?
Yeah, I mean, that goes back to the standard first question, what's the best endpoint, right? And I agree with what has been said earlier. We're discussing about how many cccDNA molecules per cells are there. Is that really important? Is it not important to inhibit progression of the disease, right? And if we want to do that, what is the endpoint that we need to measure? And I think that is a question I'm happy to put back to the whole team here. Let's discuss about that.
And just to add that we saw about 50% of the responders from Be Clear having anti-HBs. So I don't think we know at the moment. As the data mature, we'll be able to compare those that maintain response versus those that don't maintain response. And those are the exact sort of questions that we'll be asking within the clinical trial data.
Yeah, and maybe we can also just underline on this, the second we have Oliver and also here. I mean, I found very interesting the point of the observation that you might have actually this ILT elevation flare that seemed to be more linked to associated with patients that had an HDV RNA rebound and S antigen level quite higher. Of course, as the Delta situation is much more complicated, we might guess that you had a new spreading event that are facilitated if you have more S antigen comparison to very low S antigen. I don't know. What is your opinion about that, Oliver?
Yeah, that's something we're, of course, carefully thinking through. And these ALT elevations, to the extent we've seen, it came a bit as a surprise, we have to say, while we were doing the study. And we didn't expect that. Of course, there could be different reasons for this ALT elevation, and I need to reiterate, this is not seen in mono-infected patients. It's really a Delta-specific event, where one of the ideas, of course, would be by blocking S antigen and the release of infectious particles, we might have an accumulation of RNPs in the hepatocyte, which potentially could lead to hepatocyte cell death, which would be in line with apoptosis marker we're measuring in the serum. However, we heard earlier today that you could also argue that by blocking the release and an accumulation of the large delta antigen in the cell, you should have a reduction of viral replication, which goes a bit into against the theory that we have an accumulation of RNPs since replication is continuing, the particles can't leave the cell. So these are the different theories we're looking at, but for the time being, we have no good explanation. And any suggestions from anyone, what might be the underlying reason, very happy to hear about that.
Yeah, I mean, I just want to comment quickly on this ALT. Yeah, I know we have a couple of comments, but it's kind of counter to what, when you see ALT elevations in mono-infected patients, the S antigen goes down and it looks very nice, but it's the opposite really here in the Delta patients, right? When the elevation goes up, the S antigen is not really changing in these patients. Is that correct? Is that what I remember?
Yeah, I mean, we have to be careful. These patients are still on the nuke treatment. So the siRNA treatment, so the S antigen continues to decline or doesn't change a lot during the ALT flares. We see, of course, an increase in delta RNA, and we see increases of HBV DNA, which again support the idea that hepatocytes are dying, and we see a release of potentially non-infectious particles. Now, what we did indeed not observe that this would be beneficial flares. We did not see, that was initially what we were hoping a bit when the ALT increased. We saw increase of RNA and HBV DNA. We were hoping that if this ALTs continues, we should ultimately see a reduction of these markers, since the infected hepatocytes might be removed as part of during this ALT flares. But we did not observe that, so we had patients with very prolonged ALT elevations where we then had to stop treatment.
Briefly, comment from Julie before we get to our questions.
Actually, I have a question. In terms of disease, were the patients with ALT elevation had more advanced disease? That would be an explanation.
There was no correlation with disease stage. We had 5 patients initially enrolled which were compensated cirrhotics. Out of these 5 patients, 4 had ALT elevation, and that was again consistent with the S level and the HDV RNA level. Importantly, the ALT elevations in these patients with compensated cirrhosis were not different from what we saw in patients without compensated cirrhosis. There was no relevant increase of other liver markers besides ALT.
Finally, the audience. Yes.
May I directly contribute to this discussion and give a possible question that may resolve that issue? I think one of the important things we should need to know is whether the ALT elevations are related to de novo infection of cells with delta. That would mean that you would rescue or prevent these ALT flares if you would add bulevirtide or an NJ inhibitor. I think it's very important what Vir is doing here, because they have soon data maybe on a combination of an IRNA and an antibody that prevents de novo infection. And for the possible mechanism, what could be, and we should keep that in mind, chronically delta infected patients have elevated bile salts. So there's engagement of envelope proteins, subviral particles to NTCP. If you reduce this by siRNA, you may induce you may release a negative effector, subviral particles, on the efficacy of de novo infection. And that could be a possible explanation. You take away the negative regulator on NTCP, the subviral particles, and you have maybe a higher infection rate of the remaining delta that's there that provokes an ALT elevation. That's also an aspect that should be generally taken into account what you do with the S downregulation. You do something on the subviral particles that may have a regulatory effect on persistence. Yeah? Just as an idea, or some ideas that may be related to that, but we will see that. It will be interesting, of course.
Next question.
I think it's Wu Feng.
Oh, okay.
Feng.
You were before me.
Long time there.
Well, I like go back to HBV. So the question I have is regarding SMC56. You know, I think, Lena, you saw nice kind of recovery after you stopped the treatment. But in my study, I think we're talking about a couple of weeks. So the question I'm wondering, if we treat it like half a year, keep the suppressing, allow the SMC56 come back for half a year, what might happen?
Well, that's a good question. Well, maybe the hope is that maybe
I think the little clinical data, as I think Oliver, J&J compound, they have done long-term treatment, and I think there's a lot of patient data showing the rebound phase is also quite interesting. Some comes back fast, some kind of go very longer. I think in general we would believe that the compound is gone, but why the rebound just takes so long? Whether the SMC56 has done something during the half year or the longer period.
Well, as we discussed before, maybe there's also a natural decay of cccDNA molecules going on, and then after such a long-term treatment, we will also have less cccDNA, and of course, a part of the cccDNA still being silenced.
Actually,
sorry, I don't know more.
No, I had a question to kind of follow up on this, particularly for the companies doing studies, because when you show the SMC56 degradation, you show it at the protein level, right? I mean, what happens to the transcript of these? And is it something,
Nothing.
Getting biopsies is hard, but is it something we might be able to measure in like phenylelasparates or something to see if this mechanism is happening in the study?
Yeah, unfortunately, this degradation happens on the protein level. So the complex falls apart when one of the components is degraded, but on the mRNA level, nothing changes. So there's still mRNA produced. That's probably why it comes back so quickly.
That's irritating.
Massimo?
I go back to the question of the persistence of silenced or inactive cccDNA. The concept of silencing has came in now in the field of cccDNA, but we have to. It's difficult to draw firm comparison with what happens on host chromatin as compared to the mini chromosome. But we have to think and to keep in mind that the level of suppression of transcription that is behind the concept of silencing is different, and can be different in each cccDNA molecule. You proposed a very interesting concept that there are no epigenetic suppressive mark on the cccDNA under the condition you were studying in the mice, and you had a kind of compaction from the chromatin studies in the host genome. We know that many inactive regulatory regions are just actually only deserted, so you have not the positive actors. They are just not there, but they are ready to go for transcription. You have even regions that have positive and negative marks at the same time. Imagine how many flags you put to change the state. You have to remove some of these flags. So the level of suppression is may be completely different in different silenced molecules. And unless we go into this more deeply, we will never know why sometimes you have a rebound more easily or not. When you are in the liver, you may have hepatocyte with multiple cccDNA molecules, others that have only one. You cannot have less than one. Mathematically, yes, but not in the reality. So it's important to know the state. The real problem is technology. At the level of very few molecules that you have in the liver of people that are on long-term treatment or suppressed or probably under long-term siRNA treatment, the real problem is how to go into it and be sensitive enough to be able to characterize this. In vitro is easy. Probably in the mouse is possible. In the patients to do for the translational studies, I still see some limits. Most of the patients that are E-negative or that are on long treatment, they have very, very few molecules. I saw very few studies that were able to characterize the level of suppression, and to lock completely, probably you need the methylation of the DNA. We actually have no real data from patients on these kind of things.
Yes, and just to comment is that you're right, Massimo, it is challenging to measure the cccDNA, to measure the epigenetic status, but it might be that in patients like antigen negative, actually there is already a tendency to be less active of the cccDNA. There are data in this way, so it might be easier to maintain suppression maybe in this patient. That's more or less what I think, right? you are claiming, but maybe it's the claim. We need data. We need at the end some pilot study in patients. Yeah. And just to conclude on the CCCNA part, the virology, yes, still, I think we don't know that well about the half-life in this type of situation. So this is also something very important. So we know we need probably to protect hepatocytes from infection, to maybe to silence. We heard Matteo about co-stimulatory molecule that might be needed. So what about what we have learned so far from RNA interference strategy and the type of response we might get? So maybe somebody want to add in this direction?
Yeah, I mean, we've only a couple more minutes. I mean, you know, so bepirovirsen has been shown to sort of have a dual mechanism of action, so it's maybe some sort of a TLR8 stimulator. I mean, Florian, you showed it some, the siRNA. There was a couple of patients that had an ALT in Oliver, you know, in the mono-infected patients. You know, this ALT elevations are much more mild in the siRNA. I'd like to get your, well, two things. One, thoughts about this sort of maybe innate mechanisms that are associated with the different classes of drugs, ASO or your siRNA. And then, you know, it was brought up in the first session, you know, these drugs have been in the clinic for 7 years now, and we have maybe 1 or 2 week studies on T-cell responses. What are the plans? I mean, you know, to measure immunity, and what have you done now in the mono-infected patients? Let's start with. So maybe Shay, you can start from your end.
Yeah, thanks, Adam.
Oh, yeah.
Yeah, this is something we're unpicking with the data. We presented some preliminary data this year at EASL looking at the cell-mediated immunity that we see with bepi. And it's very complex and heterogeneous, as you would expect with chronic hepatitis B. We do see some patients with very robust, you know, polymerase-specific CD8 T cells. We see core-specific CD4 T cells, but it's really difficult to unpick the effect of that. So it's a work in progress. We also have the B-Fine fine needle aspirate study, which will allow us to take a look into what's happening in the liver, you know, the immunostimulatory effect. So I think that's something we're really looking forward to publishing next year.
Yeah, I couldn't agree more. I mean, different companies now have different siRNAs against HBV in development, and we see, so we know a lot about these drugs and how they act. Vir, on top of the siRNA, has an antibody that has a possible immune component to it that we need to investigate and understand more, right? We're getting efficacy data right now, but we also need to understand the mechanisms on that. And for that, T-cell activation PBMC data is super important. We are doing that in our trials, but it's slow. It's difficult and slow, which is not an excuse. We also realize that it's helpful not to do this in a large phase 2, but in smaller single center investigator-led studies, which we are initiating now. Advantage of that is that you have an investigator who knows exactly what they're doing. And this is being started now for both siRNA and antibodies, and hopefully this will lead us to the data that we're needing to actually show this immunostimulatory effect. And on top of that, of course, we are also investigating preclinical models as far as they are available.
Can I just add one very quick point? The other thing to note is that we have the Be Together study, which is the bepi followed by pegylated interferon in sequential therapy. And I think we'll understand a lot more about the heterogeneity in response, the added immunostimulatory effect of pegylated interferon. So that will be presented at AASLD, hopefully.
And Oliver, do you want to chime in?
Yeah, just in terms of data generation, immune data generation. We have done a wide range of studies with different combinations, siRNA plus a nuke, but also with interferon. Study ongoing with anti-PD-1 and therapeutic vaccines. And we are, of course, looking into multiple biomarkers. The challenge being the typical one, of course, we can look in the periphery where we can get a lot of samples, a lot of material, but it's not necessarily reflective of what's happening in the liver. We have done a couple of dedicated study, or one dedicated study where we take core liver biopsies as well as FNA samples. We have presented baseline data from that recently, and we plan to present on-treatment data for that as well, which I think will give a nice first idea of some of the data we have generated in terms of viral and immune markers in the liver as well as the periphery. And in other studies, we have implemented FNA sampling. However, as mentioned before, the results tend to be very variable. We have a high diversity of patients and responses and markers we see. So as we have seen also in non-interventional studies, I've learned over the many years in hepatitis B, still as a large variability of response and markers we see. And we have still, in this natural disease setting, still not a very good correlate of a response in terms of functional cures, or what is the correlate in terms of immune control in those patients.
Okay, thank you. I think maybe the last minutes, I don't know if we are now mentioning more and more combination therapy. So of course, try to get free of some kind of thinking about what type maybe of combination therapy you can imagine it with all what we heard somehow when we know that the collected so far could be better, kind of combination, add on. There is a feeling, nobody knows the answer, but if you just could be entirely free to think about, decide, what's your opinion on that?
I mean, that's the $1 million question, right?
Yes, what? I know.
$1 billion question. Well, the data that I've shown today shows that bepi on its own may be sufficient for some patients, particularly those with lower surface antigen. We know that with the emerging data, bepi is more effective when you've got lower baseline surface antigen. So if there is a way to reduce surface antigen, prior to receiving bepi, that would be one strategy. And then in terms of increasing the effectiveness, is there a way that you could enhance the immunostimulatory effect? So we've got a targeted immunotherapy research arm that we're looking at the impact that the additional immunostimulatory mode of action would offer in a combination type approach. But we're very aware of the overlapping toxicities, so we need to do this in a controlled way through robust clinical trials.
Okay, very short. I mean, my very personal opinion is yes, combination is needed. I don't think there is a single drug that can lead to functional cure right now. Maybe in 10 years from now, we have a new cool mode of action. So the VIR strategy is called stop and clear. Stop by meaning reducing antigens by either siRNA or antibody, and then clear by restoring immunity. Could be via the Fc gamma effectors of the antibody, could be via interferon, or could be by some other immune stimulatory compound.
On that side?
So, as I mentioned, I will talk about HDV patients. So I think we really should try to combine both siRNA against HBV and HDV to see if it might improve. And I would even go for AZO because that might also stimulate the immune response. So I think that would be really nice to try that.
I agree with what has been said before. Combination therapy is probably necessary, and but I don't know which kind of combination. I hand it over to John.
Yeah, I don't think you're going to get any disagreement that combinations are going to be needed for at least the next decade. And speaking as probably the single worst immunologist in this entire room, I can't conceivably imagine a means in which we could finally finish digging HBV out of those cells without somehow or another getting the immune system to wake up and move it forward. And because I'm such an inept immunologist, that's your guys' job.
Thank you very much. So I think we are really now managed to be over the time, but we have a kind of consensus. it will be a long, probably a long wait to reach, but we are improving. There are a lot of technology, a lot of tools also that some years ago we were dreaming of having. And still, I think it will be necessary to have a comprehensive analysis in the blood, wherever possible, a little bit in the liver, to try to understand and to design better therapy. So with that, something to add, Adam, or we close? Okay, so I would like to thank all the speakers, the audience for the lively discussion, and of course ICE-HBV and the sponsoring of ICE-HBV for allowing us to have in this symposium today. And I think we are looking forward for an update at some point. Thank you very much for coming.